The second class of organic biological macromolecules is the class of proteins. So this is our second class. First was carbohydrates, and proteins are another class of macromolecules, large biological molecules that are organic and are analogous to beads on a string. There are many more different types of proteins than there are carbohydrates or any of the other types of macromolecules. And I've got a table that lists the type of protein on the left and then a generalized description of their function in the center here. Some proteins are enzymes, which speed up chemical reactions. Some proteins are part of the structure of, say, a cell, or even outside of the cell as part of the structure of an entire tissue or organ. Some proteins are involved in storing other molecules. For example, proteins can store amino acids for the production of later proteins. Ovalbumin in egg white stores proteins for the developing embryo. And protein in milk is a storage protein. There are also storage proteins in plant seeds, all of this to provide resources to the growing plant. Proteins can carry things too throughout the cell and throughout the body. For example, hemoglobin is a protein that you find in red blood cells that carries oxygen to all the tissues that need oxygen in order to create energy. Some of our hormones are proteins. These are called peptide hormones. And the job of hormones is to coordinate a whole organism's activities. So for example, insulin is a hormone that your pancreas secretes when blood sugar is high. And insulin targets different tissues in your body, including liver tissue, muscle tissue, to store up glucose for later as glycogen. So examples of two carbohydrates that we talked about in the last lecture. Some proteins are receptors. They're found on the outside of the cell, and their job is to receive some sort of signal, which can actually also be a protein, from another cell. And that also coordinates the organism's activities by causing the cell to create some kind of response. For example, you have receptors in your nerve cell membranes that accept nerve impulses in order to allow your muscles to react. There are receptors on liver cells and on muscle cells that receive insulin so that those cells then take in glucose and store it up for later. Many proteins are involved in movement, either cell movement or whole organism movement. There are proteins on the outside of single-celled organisms that swim through their environment, aqueous environment, and actin and myosin in your muscle cells work together to contract your muscles, and that aids in your movement. And then last, there's a whole class of proteins that are involved in defense, your immune system proteins. Antibodies are proteins that your white blood cells make to combat bacteria and viruses. So we've got this generalized list of types and functions of proteins. But the human cell can actually make as many as 100,000 different types of proteins. And all of these proteins would then fall into some specific type of protein with a specific function. Each one of those 100,000 has a specific function, and it can't do anything else. So every protein has a job. Now, some of those proteins you might find in all cell types because their job is required in all the different cell types, liver cells, heart muscle cells, any kind of cell. But then there are other types of proteins that cells make only in specific cell types. And you're going to see later that even though you have the instructions for making all 100,000 proteins in every one of your cells, there are plenty of proteins that only get made in specific cell types. For example, insulin is a protein. It's that signaling protein that the pancreas makes in response to high blood sugar. Only the beta cells of the pancreas make insulin. So that's not a kind of protein that you're going to find functioning in another type of cell. Then there are proteins that are involved in making energy. Those proteins are present in every type of cell because every cell needs energy. So there are many, many, many proteins, 100,000 roughly. Some work in all cell types, but others are specific for one specific type of cell. Now, all the proteins have the same basic construction, We can show them as analogous to beads on a string. So the protein is a polymer built from monomers, and the monomers are amino acids. So even though they have the same basic construction, every one of those proteins is going to be different. And the difference is the length of the amino acid chain and also the order and type of amino acids in the chain. Let's take a look at the structure. This is the structure of the bead, an amino acid. An amino acid has one carbon atom which is covalently bound to 4 things, another carbon that's part of a carboxyl group, a nitrogen that's part of an amino group, hydrogen, and then also a chain of atoms known as the R group. And it's the R group that makes every amino acid different. There are 20 amino acids, and all 20 of them have the same basic structure, but it's this R group or side chain that makes them different. We can categorize our 20 different types of amino acids into 2 groups, hydrophobic and hydrophilic. So the backbone of this chain of amino acids is always going to be hydrophilic. There's a carboxylic acid, or excuse me, a carboxyl group, an amino group, and then the side chain just determines whether or not the entire amino acid is considered hydrophobic or hydrophilic. Notice that we've got all carbon and hydrogen here. That makes it hydrophobic, all nonpolar covalent bonds. We've got a hydroxyl group here. We've got a carboxyl group here. These 2 types of amino acids are examples of hydrophilic amino acids. There are also amino acids that have like an amino group down here. Remember that a carboxyl group and an amino group both become charged in solution. So they're hydrophilic not just because there's something polar involved in the side chain, but because they actually have a charge to them. And remember that hydrophilic is anything that is charged or polar, has some sort of charge, even if it's partial charge. If it does, then it's attracted to water. So this is going to be important when we look at how proteins fold up, because it's their folding into a particular shape that determines their function. So here we have 2 monomers, 2 amino acids. They're very generic because the letter R is used here to denote the side chain. And when we put 2 amino acids together by dehydration reaction, we release water, but we form a bond between the carboxyl group of one and the amino group of another. This is a covalent linkage, and it's called a peptide bond. So just to think back, the covalent linkage between 2 monosaccharides was a glycosidic linkage. The covalent bond between 2 amino acids is a peptide bond, and here we now have a dipeptide. There aren't dipeptides doing anything in living cells. A protein actually consists of many amino acids linked by many peptide bonds. So the long chain of amino acids that we're going to look at is then called a many peptide for peptide bonds. So imagine when we look at the protein, which could be strings of hundreds of amino acids, we're looking at amino acids joined by peptide bonds, and the entire chain is called a polypeptide. Now, a protein isn't really a protein. It's a polypeptide and not considered a protein until the polypeptide folds. And the folding confers the protein's function. When a protein is folded up, it's in a particular 3-dimensional shape, and it allows it to very specifically interact with other molecules. And if it wasn't in that shape, it couldn't do its job. It couldn't interact with whatever it needs to interact with. So imagine this is a ribbon structure depiction of a protein with hundreds of amino acids. I know you can't see, they don't look like amino acids, but hundreds that are folded up into a 3-dimensional structure that provides different pockets or grooves. That groove might be a place where an enzyme binds reactants in a chemical reaction. It might be the groove or spot where oxygen binds hemoglobin, but that groove has to be there. Any groove has to be there so that it can bind whatever it needs to bind in order to do its job. This structure gives you a little bit more detail on amino acids, but I want you to notice that we still have these grooves and pits in a very specific 3-dimensional shape. Now, the folding of a polypeptide occurs at 3 levels. The primary structure of an amino acid is its sequence, the primary structure of a protein is its sequence of amino acids. So each one of these beads is an amino acid, and the name in here is the 3-letter abbreviation for the type of amino acid. And you'd never find more than 20 different types in here because there are 20 amino acids. But notice that, like, alanine is in multiple places along the length of this protein. Another protein would have a different sequence of amino acids. And if we could really zoom in on this amino acid, we'd see that the structure of each one of these is a carbon, amino group, carboxyl group, and side chain or R group. And every amino acid has every component that it needs in order to form the peptide bond and still have its little side chain available. The secondary structure of a protein is its initial folding. And the secondary structure is always hydrogen bonding between atoms of the backbone. And the backbone would be this polypeptide chain, part of the polypeptide chain that's in common between the amino acids. So it doesn't include any interactions between R groups. But this is an oxygen here, a hydrogen here. So like, for example, a hydrogen of the amino group and an oxygen of the carboxyl group of an amino acid a few away. Actually, this is probably about 3 amino acids away. And there are 2 main structures that are formed in the secondary structure, the alpha helix and the beta sheet. And we think of these as structures that are formed by relatively close to each other amino acids. The further folding of the protein is its tertiary structure. And its tertiary structure is stabilized by interactions between R groups. So imagine you take this coiled up protein and then you fold it up even further. And the folding is kind of glued together or held together by interactions that are between now the R groups, which are in yellow. So here they are sticking out. Here they are sticking out. And this protein, which is already in its secondary structure, can now fold up into its tertiary structure because these orange side chains will interact with each other. So things that are positively charged and negatively charged might be attracted to each other. Things that are polar are attracted to each other. Side chains that are non-polar are attracted to each other, not because the strength of their attraction is so strong, but their desire to be completely far away from anything polar or charged is incredibly strong, so they tend to cluster together. And this gives most proteins its final folded shape. Now, some proteins actually consist of more than one polypeptide chain that has folded up into its secondary and tertiary structure. So imagine this twice, or 3 times, or 4 times. Could be the same type of polypeptide, or it could be 2 different polypeptides. But that joining of those 2 units would be quaternary structure. And not all proteins have quaternary structure. Only those that need to have what we call multiple subunits will have quaternary structure. So for example, hemoglobin. Hemoglobin is actually a protein that has 4 subunits. 2 of them are called the alpha subunit, and 2 of them are called the beta subunit. And the alpha subunit is a totally different sequence of amino acids than the beta subunit. 2 alpha chains fold up into the tertiary structure. 2 beta chains fold up into the tertiary structure. And then the 4 of them bind together to create a tetramer, or a 4 subunit protein. Now, some proteins might have subunits that are all made up of the same sequence of amino acids. But remember that not all proteins have multiple subunits. Actually, most proteins only have 1 subunit folded up into the tertiary structure. So we're going to talk a lot about proteins. Not as much about carbohydrates, but a lot about proteins. And so I just want you to have a general idea of what a protein is. And very most important is thinking about that a protein can't do anything until it is completely folded into its proper shape. The next thing we're going to do is talk about nucleic acids, and nucleic acids, particularly DNA. There are only two types, DNA and RNA. DNA encodes the instructions for making every single one of these proteins in the cell. And that's the subject of our next lecture.